Well, good evening, everybody. It's a great pleasure to see so many people come to tonight's lecture. I'd like to welcome Keele colleagues, both academic and professional colleagues, and also Toby's sister, Lizzie, and external visitors that have come from Toby's research collaborations. So this evening's lecture then is been given by Toby Bruce from the School of Life Sciences. And I'm just going to give a brief biography before I invite Toby to come up and speak. So Toby's always had a fascination for nature and the environment, and studied biology at University College London, and during that time became particularly interested in the ecological challenge of safeguarding harvest from attack by pests, which is kind of quite a niche thing to get so interested in at such an early age. And following that, he completed MSc in applied entomology at Imperial College, and then followed that by a PhD in chemical ecology at the National Resources Institute at University of Greenwich. And then following his PhD in 2000, Toby joined Rothamsted Research, which is, for those of you who don't know, a research organisation that really pioneered using science to benefit agriculture. And Toby was there for 17 years at Rothamsted, during which time he gained university teaching experience as a visiting lecturer and successfully supervised PhD students and postdocs, which I would say is relatively rare actually in research institutes to have engaged so strongly with universities in both teaching and supervision of PhD students. Now, after 17 years at Rothamsted, Toby was appointed to an externally advertised chair that we had at Keele in chemical ecology, with the intention being to really develop a new world-class food security research unit here at Keele, specifically focusing on crop protection within our Centre for Applied Entomology and Parasitology, which is an internationally renowned centre of kind of very long-standing, within our sort of expanding and significantly expanding and thriving School of Life Sciences. So Toby joined Keele in 2017, and so not been here that long, and in the time he has been here, I think it's fair to say he's had a tremendous impact on School of Life Sciences and Keele as an institution generally, and he's a pivotal part of our new Institute for Sustainable Futures, which we launched just a couple of months ago, just a month ago. And in his first year, Toby has won 3 BBSRC grants. So if he's here 10 years, I look forward to 30 grants coming in. But it's pretty impressive. I think we obviously had high expectations from it, but I think he's exceeded those expectations. And I think that I'm sure there is more to come. I know there's more to come from certain things that we've heard recently. And so he's already started to build a significant team of postdocs and research students. So Toby's research then is internationally leading, working on agriculturally important insects, and within this work he seeks to understand their complex interactions with their host plants and their natural enemies, and tries to devise novel interventions that reduce crop losses to pests. They're hugely important in terms of sustainable futures and global health. I think it goes without saying that in appointing Toby to a chair, he'd already achieved an awful lot. So he's published very extensively in highly cited journals and papers, very strong track record in securing competitive external research grant funding, and also in building strong international research collaborations, which are obviously imperative to be able to deliver grant success in the current climate. And say he had already supervised 10 PhD students before coming to Keele, which considering he wasn't at university is pretty impressive. And I think one thing we've also seen about Tony is his real enthusiasm and desire to inspire the next generation of scientists, and we've seen examples of that. In fact, the television audience has seen examples of Toby at work, and really is one of our kind of media stars in that respect as well. So can I therefore formally welcome Toby Bruce, Professor of Insect Chemical Ecology, and invite him to give his inaugural lecture, Balancing Nature to Protect Harvests from Pests. Toby.
Well, thank you, Mark, for that very kind introduction. It's a great pleasure to be here, and thank you, everyone, for coming along this evening. So my talk is about balancing nature to protect harvests from pests. And first of all, let's see the agricultural connection in Keele. So agriculture is actually embedded within the Keele crest, because you have here a scythe that's coming from the scythe in the Sneed family crest. So there is an agricultural connection at Keele. I'd just like to draw your attention to that, but I'm trying to strengthen that with some of the new research initiatives which are going on here now. So let's start considering the topic for this evening, though, and that is how to protect the harvest from crop pests. And this is actually quite a wicked challenge, because what you've got here in an agricultural environment is like a heaven for the pests. You've got an almost unlimited supply of the food that they're adapted to feed on. It's been selected for other purposes than defence. It's been artificially selected for yield and quality. It's been fertilised, it's been irrigated, it's lush. So this is a resource, and obviously we're trying to use this for human food security. But the insects that are adapted to eat on that, they quite like to take advantage of that as well. And so that is the challenge. And if things go wrong, then it can be very bleak, because these insects, they can really wipe out a crop. I mean, we've had some quite effective crop protection, but there's increasing challenges with insecticide-resistant pests and invasive pests, such as the Forley armyworm, which we've got a new collaborative project on with Zia Khan in Issappi there. So there's a big challenge of how you protect the crops.
Any fresh growth in their path.
So in popular culture, then I think the locusts are something which generally people are more familiar with, because they're
steady munching. The infantry
but
becomes airborne.
This is one example of a crop pest, and the kind of damage that they can do if they're left unchallenged.
While the food and moisture.
But if you contrast the agricultural environments, where you've got a very simplified situation with what's happening in nature, that's very different. And so there you have a lot more biodiversity, and the plants they haven't been artificially selected; they've been naturally selected, and so they're adapted to withstand the attack from insects. And there's a huge diversity of insects on Earth. In fact, there's more insect species than any other animal. 3/4 of all animals are insects, and it's only a tiny minority that are pests. But they make up for that by their large numbers and their ability to very rapidly reproduce. And so there's a small number that, as I say, that are pests, but they make up for those small numbers by their large impact. And so the vast majority of insects, they're actually very very specialised and not so damaging. And although there's some major challenges which we're now facing increasingly in agriculture as the pests evolve resistance to the pesticides that we've been relying on, there is it's not all doom and gloom. I think there's room for optimism, because if you look at the really big picture, and we're really zooming out now, so if you look at the Earth from space, the land area where you haven't got a desert is largely green. And so the plants which have been naturally selected, they've evolved ways of surviving this onslaught of attack by herbivores, and they've been doing that for a very long time. So the early origin of herbivory was actually 400 million years ago, and over that time, the plants have evolved some really sophisticated defences that they use to protect themselves from these attacking organisms. I think we're really only just scratching the surface. I think there's a whole lot more to learn about this. And also, if we sort of look at the how we got to this situation where we are currently, for the majority of human history, we were actually hunter-gatherers. The development of agriculture is a relatively recent phenomenon. It's within the last 10,000 years. So, and as already mentioned, the agricultural environments, they're simplified and vulnerable to pests. But these crops that we have now, they've been artificially selected, and they're nothing like the wild plants that they came from. If you look at these wild grasses that the wheat, for example, was domesticated from, they've got tiny little ears, and in comparison to modern bred wheat, and over that process of human selection, these plants have been optimized, and it's really quite a remarkable achievement if you consider that that these early farmers, they had very little understanding of genetics, but they were just selecting the plants which kept providing the better growth and better nutrition. And it's really remarkable that we've actually come to these plants, and there's been a lot more development in recent years, of course, as well, in terms of the improved high yielding varieties. But I really would like to make this point. If you don't remember anything else from what I say this evening, I think that this is the point that we really need to remember, and that is that the agricultural ecosystem, it's not a natural ecosystem. So first of all, you're taking a plant and you're growing it outside from its area of origin. So to take one example of maize, it came originally from Central America. It's now grown all over the world. So you're now growing it from, exposing it to insects and various other organisms that it didn't originally evolve with. And then, of course, you've got a much thicker stand. So you've got a much denser stand of plants there. There's all kinds of differences, but the plant diversity, the plant density, the frequency of disturbance, because in our agricultural situation, every few months or weeks, the thing all gets cut. So that's quite different, and then the actual plant traits itself that's been selected for having bigger structures of the parts of the plant that we eat. And in that environment is a very unnatural environment. We can't really go back to nature, because if we did, then there wouldn't be enough food to feed the world if we went back to being hunter-gatherers, because current human population levels are just too high. And the pest situation is actually getting even more difficult, because it's been sort of propped up by the use of synthetic pesticides, but the availability of that is going down due to the evolution of resistance and the reduced discovery and approval of new products. So that means that there's fewer effective pesticides. There's a reduced genetic diversity in our crops, so there's not enough resistance traits in the crop itself. And then trade helps to facilitate the spread of invasive organisms around the world. And it's predicted that the climate change, that's going to make conditions more conducive for pests as well. So there are a lot of different factors which are making this environment really, really vulnerable to pests. And it's kind of taken for granted that we're always going to have plenty of cheap, readily available food, because that's due very much to people like Norman Borlaug, who did the first green revolution and developed these wonderful high-yielding varieties. But those varieties, their yield is conditional on having good crop protection. If you take away the crop protection, you're not going to realise that high yield. And so we've got a really serious threat here with these crop pests, and what to do about them is a major research question. If we want to keep having this wonderful supply of food that we currently take for granted. And so the subject, it might sound like a bit of a kind of niche interest and a bit of an unusual subject, but actually it's not just the health of the crop itself, and we're not just interested in the plants for the sake of themselves. It's because of their importance for human health through nutrition, that the crop is actually really important for society. And not only that, but there's also the environmental footprint. So people will often think of farming as a kind of quaint rural occupation, but it actually is the human activity that uses more land than any other industry. It uses more water than any other industry. It's vast amounts of resources which are consumed for producing the agricultural produce. And so if we could actually have more efficient agriculture and minimize the use of resources that are used in agriculture, then we could conserve more land for environmental conservation and things like that. So it's actually fundamental for protecting the environment. So taken together, it means that this topic is actually key for global security, and there's a lot of investment is made in defence for security. But actually, if you can have good food production, you can actually help to prevent wars from occurring in the first place, because a surefire way of getting people to riot is if there's lack of food security. So this is a sort of broader context of it, and there was this landmark report by the Royal Society quite a few years ago now, reaping the benefits, and they made the point that we need to have a second green revolution where we're producing more but using fewer resources, because to feed the growing demand, it's not only human population growth, it's also richer people having more resource-intensive diets. But how to meet that demand without ruining the environment is actually a huge challenge. You know, we want to be able to face that. So, and we're not going to achieve this sustainable intensification or ecological intensification of agriculture if we don't have good crop protection, because that's going in completely opposite direction if we're losing large chunks of the harvest to these pests. So how can we balance nature to protect harvest from pests? So we've got this artificial situation with a resource that's really vulnerable. And so it is actually an ecological question, and we're dealing with populations of living organisms, and that's precisely what the science of ecology is about. And so if you look at it at the different trophic levels, you've got the plant, and it's under selection pressure to evolve defense mechanisms against these attacking organisms that try to eat it, and also to attract the natural enemies, because the pests are the friends of the crop. But then for the second trophic level, for the herbivore, which is the insect which is becoming a pest in an agricultural context, they want to find the right host plant efficiently, they want to avoid unsuitable hosts, and they want to avoid predators. And then we have we could actually go higher. You can even go up to the fourth trophic level with the insects that attack these ones. But let's look at these three to begin with this evening. And the natural enemies. These are the predators and parasitoids that attack the pests. And so if we could increase those, then that could help to provide some very good biological control of the insect pests. And in fact, so some of the plants, they've evolved to release a cry for help signal when they're attacked. So they release a different smell which can attract these kind of insects. And so that's quite a fascinating thing. I'll show you a little bit more about in a few slides' time. But the selection pressures are for all of these are operating in different ways, and the insects they've all evolved sophisticated olfactory systems for detection of their hosts and avoidance of non-hosts. So it's also for these predatory insects, they also need to find their hosts. So these are the research questions. At the first trophic level, the plants and the herbivores, what makes a host plant suitable for the herbivore, and conversely, what makes a non-host unsuitable? That might actually be even more interesting, because if we could get some of that and put that on, get that transferred to the crop, then it might solve some of these problems. And how can we make the plants less attractive to pests? And then on the second trophic level, the third trophic level, the predators. Then how can we increase the attraction of the natural enemies? And so the signalling between these organisms. That's the subject of chemical ecology. So it's a subject with a very broad remit. It's actually wider than just the crop pests. It's actually all kinds of signals between living organisms and how that works, and the chemical basis of that signaling. And as well as identifying the chemical structures of these compounds to really understand how this works, then we also need to understand the context of it, because sometimes, as you will see, these chemicals, they can have different meanings to the animals in different contexts. But the insects, they've evolved some remarkable sensory abilities, and if you think that in a wild habitat, sometimes they might be finding a rare plant and they need to be able to pick that plant out amongst many other things, and the olfactory system that they use for that, it provides them with a simplified representation of the external environment, so that they can find the resources that they need to maximize their fitness. But perhaps it's not that simple, actually. When we look into it, it turns out that it's actually pretty sophisticated, and the insect, she can map out the volatiles and these smell compounds in multiple dimensions of space and time. They can process complex blends and detect very subtle changes. And they're not completely hardwired. There's some cues that they may be hardwired to, but a lot of these cues they can actually learn from previous experience and through associative learning. And so if the cues that are associated with rewards and that improve fitness, they respond better, and conversely they avoid things which could be associated with a threat. And this better sensing that they have, and it's really a very sophisticated system. It has the selective advantage to enhance their fitness in finding mates and food. And although it's evolved over an incredibly long time period of 400 million years, the insects are operating in very fast-paced environments, and they have to make split-second decisions. For example, with an insect deciding whether to colonize a host plant, should I land here or not? And the way that we think this works is through a very fine spatiotemporal resolution of these signals. So here you can see the neurons, and just to sort of try and explain it, just to say we've got two chemicals, A and B, that they're smells which are associated with the host plant. And here you have this when these things fire faster, it's when they're getting excited. So this is time over here, and the number of spikes shows when it's excited, and when they come closer together, it's responding. And so if there's simultaneous excitement of these two olfactory receptor neurons at the same time, then these two chemicals, they've occurred together. And this is just to simplify A and B. It might actually be more than, it might be 5 or 6 of them. But if the insect can detect them at exactly the same time, we're talking about a matter of milliseconds, it can differentiate that from when it's getting them at different times, where you might have non-host plants that are also producing those chemicals. And we did, a number of years ago, a review of chemical ecology studies from a number of labs all around the world, just to see what was the most prevalent type of coding mechanism for these host signals or host cues, if you want to call them that the insects respond to. So here we have the plant volatiles. This could actually, it also applies to the host finding of predatory insects too, but it activates an olfactory receptor neuron. So ORN is the olfactory receptor neuron, and CNS is the central nervous system, the brain. And so there's two contrasting hypotheses, and they're not actually, it's not necessarily one or the other, because there could be some examples in some situations of one and other examples in another, so it's not completely just one or the other. But the two alternatives are that you could have very taxonomically characteristic plant compounds or host compounds that the insect responds to, that only occur in that plant and not in another one. Or you could have much more commonly occurring compounds, but it's like a combination lock, and can you get these particular ones occurring together at the same time? And it actually, it turns out that the most frequently occurring one is actually the second one, this blend recognition. But there are a few exceptions, like the isothiocyanates, which are characteristic of brassicaceous plants, that the specialized insects that feed on those like. But this seems to be the most commonly occurring thing. And there's a wonderful example from previous PhD student Ben Webster, and he did some electrophysiological recordings from the insect 1010 using black bean aphid, and identified these electrophysiologically active compounds, the host compounds, from bean plants. And the really interesting thing about it was that if you expose the insect to these odors in an arena where it's just got one presented on its own and not outside, without the context of the blend, although they're coming from the host, they actually repel them. So when you can see these bars going down negatively, it's when the insect is repelled in this olfactometer device that we call it. And if the line goes upwards, then there's a positive response and it's being attracted. And these compounds, they're all done at different doses, and these are the doses that are most repellent. And then what Ben did next after that, he took them, and we thought it'd be really interesting to see what happens if you put them together as a combination that would be more like what you get in a host plant. But these weren't the natural concentrations. They were actually the concentrations that were most repellent in dose-response studies, because he was seeing if you could find another dose that was more attractive. But it actually turned out that there were other doses that were more repellent. But if you put them together as a mixture, and this really shows you why it's so important to have this mixture for conveying the message. It's not just the chemical structures, it's also the context. Because if you put them together as a mixture, there you are. You've got the opposite response. It's become attractive now. So, you know, this combination of compounds which are repellent by themselves, you may intuitively think that if you put them together as a mixture, it becomes a super repellent. Well, no, you get completely the opposite behavioral response, and so it suddenly becomes attractive. So that shows how important it is to have these mixtures of compounds for conveying the message to the insect. And if you actually think about it, it makes a lot of sense, because in nature you wouldn't get those odors being just coming one at a time. You get them as a mixture. But anyhow, that's the subject of chemical ecology, and a fascinating subject, and we're hoping that we can try and exploit that to develop some better approaches for managing crop pests. But now we want to sort of try and bring it back on a more practical direction. So thinking about resistant crop varieties. So this is the first trophic level. Can we do something about them to improve their resistance? And there's a super example of this to show what can be done with the orange wheat blossom midge. And so this is a very specialized insect. It feeds on wheat and a few closely related grasses. But it's a very pretty female insect, but she lays the eggs, and the larvae emerge, and they exude these enzymes from the body wall, and they digest the grain, and it's just precisely in the economically important part of the crop, and it causes all this shriveling and pre-sprouting. It damages the grain, and it facilitates secondary fungal infection. So it's a very nasty pest. And in 2004, there was an outbreak of it, and about 10% of the UK wheat harvest was lost to it. So it was causing a lot of damage. But it varies a lot from year to year. But this pest is now much less of a threat, because we had a big collaborative project on this, and one of the outcomes from that was these orange wheat blossom midge resistant varieties, and they're still attractive to the females. They still come along and they lay their eggs on them, but the larvae don't survive. And what happens is it forms a wound plug there, where the larva, the grub, the insect baby, is trying to feed, and they don't survive on this, and so we think it's a kind of lignification process which is going on there. But one thing that's very, very clear is how effective this is. So this is the very first data that we got from that project, and these are different wheat varieties, and you've got the level of damage up there on the y-axis, and most of them are heavily damaged. At that time, the option and tanker were very popular varieties, but the farmers were inadvertently growing things that were highly susceptible. But this was the first resistant one. And the resistance is really remarkable. It's very, very low damage on that. It's pretty near 100% resistant. So, and this makes life easier for the farmer, because if you just grow one of those varieties, you don't have all the bother of checking your crops and putting your pesticide sprays on. They much prefer to have that if that's available. And it's now been bred into about 60% of the UK wheat. It's now resistant to that pest. So that's fantastic. And I mean, here you can see it on the recommended list that farmers use for choosing which wheat variety to grow. But it's just as well, because the main insecticide, the most effective one for controlling this pest, was banned in March 2016. So it's just as well we've got something else for that pest, although there's a number of other pests where the situation isn't so fortunate. And another thing that was developed for orange wheat blossom, which was the pheromone monitoring traps that allow very sensitive detection of when the insect is there, because it varies a lot from year to year. 4 out of 5 years, it's not going to be a problem, because it arrives either too early or too late. It just comes out in a flush, depending on the weather conditions of when it comes out from these overwintering cocoons in the soil. And by using these tools, then the farmers, they can avoid unnecessary sprays, and so they only put the insecticide on when it's necessary. So this is using the sex pheromone of the midge. So it's the compound that the female releases to attract the male. And it's very, very species specific, so all these insects in there. And it's very, very sensitive. They're all orange wheat blossom midges, and so that gives an early warning of when the pest is around, and to tell you if the fields need treatment, and then you can relax if they don't need treatment, because if these traps aren't catching, then obviously it's not going to be a problem. And we developed a kind of decision support tree to make it straightforward for farmers and agronomists to use, so with all these yes-no decisions and different thresholds. But of course, if you're growing a resistant variety, you don't need to worry about all of that. But then, that's a nice situation, and we were fortunate to find the resistance in an already commercial germplasm. But there was another project where we had on the bean seed beetle, and over 600 different bean varieties were screened, and unfortunately none of them were resistant. So it just depends on what you have there in your gene pool, because it can be a lot of hard work and nothing out of it if you don't happen to have the resistance there. Something more recent that we're looking at, in fact, got a PhD student, Jamin, in the audience, who's looking at this right at the moment, is these wild potato varieties. So these are Solanum stoloniferum, and we've got some early data from that showing that there's very low aphid survival on these resistant wild potato lines compared to the cultivated Desiree potatoes. So we're still trying to work out the chemistry that's involved in that. But when you start trying to get resistance from a very distant source, then as has now been found, actually, my colleagues at Rothamsted, they've involved in this very big wheat pre-breeding program where they screened hundreds of different wheat varieties, but the only ones that are really resistant to the kind of wild grasses that the wheat was domesticated from. And so this has got aphid resistance, and this is actually a picture of a couple of ears that I pulled up in the field. And sure, it's got the aphid resistance, but the challenge is, how do you get that resistance from that wild source? I mean, you want to rewild your crop and get that trait that makes it resistant, put it back into this bred wheat, which has got much, much bigger grains. I mean, you're not going to get much of a harvest, and it's not going to taste very good if you try and make bread out of that. In fact, it's going to be awful. It's going to be going back 10,000 years in the history of humanity and have food crops. But so that is the challenge. How can you get that? And that challenge could, to a considerable extent, be solved if society was willing to accept some of these more sophisticated plant genetic improvement techniques. And so, if you want to take a gene from a wild potato and put it into a cultivated potato, you could actually do that very quickly. So this is images courtesy of Jonathan Jones at the Sainsbury Laboratory, and this is his field trial, and this is looking at blight resistance. It's not an insect, it's a disease. But this is the control plot of the untransformed potatoes. And they got really hammered by the blight that year. It was a very bad year for the blight, and there's no potatoes left there. It wipes out. And I mean, normally what would be done, you put on, this is actually conservative to say 10 fungicide sprays. It would probably be more like 15. But that's how it's normally protected. But that didn't have any pesticide treatment, and it was just exposed to the disease. And then this is the one which has got the resistance gene from the wild potato. and it's actually stood up to that really heavy attack. But then this is the question, how do you look at that? Is that transfer of genes? Is that rewilding your domesticated plants, or is it something which people, a bridge that society doesn't want to cross? But I'll leave that debate for now. But that's something to consider, though. But I was involved directly myself in a project with genetically modified crops, and we were looking at for this to see if we could use that for delivering some of the chemical ecology. So a lot of these volatiles, they're unstable and they're difficult to formulate, to get long release of them. So often we have these little plastic dispensers and things like that. So we thought, well, why can't we use a green chemistry approach and get the plant to make it for us? And we picked on the aphid alarm pheromone. When an aphid is attacked by a predator, it's got this sticky secretion from the siphunculate, but there's also a smell that comes out from that, and that's the alarm pheromone that makes other aphids run away. And you can see here there's a little drop of it, there's a solution of it there in the syringe there. And there's actually just a tiny amount of it. But as soon as the aphids smell that, I mean, they're plugged into the plant, they're the green fly. They don't move around very much, they're just plugged into the plant. But as soon as they smell that, they're off, they're running away. And it's because they've got this as the warning signal that they use to tell their neighbouring aphids when there's a predator around. And so that was really, really promising in the lab, and so we thought, well, what happens if you engineer that into the wheat? So the wheat is making this repellent odour, and it was possible to do it. We had a collaboration with Hugh Jones at Rothamsted, and he engineered the wheat, and it very successfully produced this E-beta-farnesene. And the nice thing about wheat is it's not a very smelly crop, and so it's similar to the pure E-beta-farnesene that you get from an aphid when it's being attacked. But so it's very successful in terms of being able to release the chemical and get the plant to make it. But perhaps in retrospect, looking back at this, perhaps we chose the wrong type of semiochemical for this, because because in nature it's released as a very short, sharp burst. It goes up over time very quickly and goes down over time very quickly. But these plants, they're continuously making it, so all the time they're just pumping out this odor, and they did that very well. But if you do short-term with these simple olfactometer bioassays in the lab, you can do short-term bioassay. There's very good repellency, and it's very, very promising. But if you look at the aphid populations in the field, they weren't any different. So it's a difference if they got a long or a short exposure to it. The other hypothesis we were testing in that project was that because the parasitic wasps, they use that aphid alarm signal as a foraging cue, so they use that to find their prey. And so could we increase the parasitism of the aphids and have more of these aphid mummies in those transformed plants? But there wasn't any difference in that either, so the insects could tell the difference. But now at Keele, I haven't been here so very long, but one of the things we've been looking at is the prospect of using improved formulations for phytochemical treatments. And so, as we've discussed, a lot of the crop plants, they've lost the phytochemicals that exist in wild plants. And so the idea here is to identify some of those chemicals from the wild plants and then have a sophisticated way of applying them to the crops. So we're exploring nanotechnology and also complexing of molecules to improve the formulation of it, so we can apply those to the crops. But this is quite an early stage, but there's all kinds of different phytochemicals that exist in nature. So there's antibiotics that can have contact or fumigant action. There's antifeedants. There's things that can interfere with the insect reproduction. There's repellents, and there's also attractants for natural enemies. And so we're doing some simple bioassays using aphid Mysis persicae as a model system. And this is looking with a conventional hexane formulation at the aphid mortality, and there's not much difference between treated and controlled with these. We're just using as a starting point some essential oils as a source of plant secondary metabolites. But if you have a nano formulation, then that improves the performance of it, and so we get 100% mortality of these aphids in this particular experiment, and so it's provided a really good, not just repelling them, it's also killing them. And looking at repellency, you can also improve repellency, but when it's got this polyallylamine nanotech formulation, so we've got improved, much stronger repulsion in the olfactometer compared to the standard hexane formulation for that particular plant extract there. So there's also some activity there with the peppermint. But, so that's looking at the plant, and so I think there are a lot of opportunities to try and make the plant more resistant, and we can learn from nature for that too. But the other prospect is, what can we do to improve biological control? So can we get more of those wasps, that, for example, that attack the aphids and form these aphid mummies, and then the wasps emerge? And there's already been considerable progress with this in greenhouse environments. So in the greenhouses, there's a release of large quantities of these, but the difference in a greenhouse is that you've got a roof, and so if you release these insects, they don't move off so much from where you've released them. But in an open field situation, it's more difficult to do that, and because the insects can disperse. But what we tend to have there is conservation biological control to try and make the habitat more conducive for these natural enemies. And so here you see the greenhouses, and this is an Almeria region in Spain, and the whole landscape is covered in these greenhouses. And they've largely replaced pesticides with the use of biological control in those environments. It's been very successful, but the next frontier in biological control is the open field environments. And so can we do some things to slow down the pest growth rate and increase the speed of arrival of the natural enemies? So that's one of the things to look at. And there's actually has been some progress in terms of the artificial release of natural enemies in open field environments too. So in Brazil, they're doing these very interesting experiments. Well, they're more than experiments now. That's gone through to commercial production, and they're mass rearing the eggs of this parasitic wasp, the trichogramma, and then they're getting these drones to release the eggs in the fields. But it's really interesting to see how that's going. But in open field environments, though, it does tend to be a bit more like conservation biological control. So you've got this habitat which is almost like a desert for these natural enemies. And how can we improve make it more attractive to them and try and get them to come out into the main crop? Because there have been quite a lot of developments that are literally tinkering around the margins. But these insects, the beneficial ones, they're so happy in the margins they don't penetrate deeply enough into the main crop. So could we use some kind of semiochemical to bring them in there? Or improve the conditions for it. And so there are some certain things like that. So, for example, the cisjasmone treatment. So when we treat plants with this plant activator, we can double the amount of time that these wasps spend on the plants. And so, and that has helped to reduce aphid populations in the field. They have direct effects on the aphids too. So it reduces aphid settlement. It makes the plants less attractive to the aphids, so it switches on the plant defense. And this is actually field data, and we've found a significant reduction of aphid settlement in the field there, so that was encouraging. But if you look at the model plant Arabidopsis to try and see some of the genetics underpinning this, there's one gene that was really important for this. So there's the CYP81D11 gene, and if you overexpress that gene in the plants, then you have the plant you don't need to treat it with the cisjasmone. It's constitutively attractive to these natural enemies, so they're spending significantly more time on those plants. So that's the cisjasmone. But then there's another story that's really fascinating with the attraction of these natural enemies, and this is actually another PhD student project with Zdenka. She was at Aberdeen at that time, and she was testing a hypothesis that the mycorrhizae underneath the plants, that they could function for signaling attack to neighboring plants. And so it's very well known that when a plant is attacked, it can become more attractive to the natural enemies, but more repellent to the pests. But could it actually do the same thing to the neighboring plant? And this was the experimental setup. So there's various treatments. So you could rotate these soil cores, or you could have a very fine mesh that would prevent the hyphae of the mycorrhizae from penetrating through. And she collected the odours from these various different plants, with and without a mycorrhizal connection. And the donor plant, by the way, in the middle, that's exposed to aphids. So that's the signalling plant in the middle. And what happened was that the odours of the plant that had the aphids on, as expected, it was repellent to the aphids. But also, the plants which had this mycorrhizal connection, they were also repellent. So that signalling had worked, and so they would significantly repellent the odours of those plants. But if there wasn't any hyphal connection, then the host plant odours were still attractive. So that's with the aphid. But often in chemical ecology, what we see is that the signals that are repellent to the pest are attractive to the natural enemy. And the reason for that is because it's associated with a plant which has been attacked. And so the natural enemies, they're tuned into that for finding their prey. And so with these parasitic wasps, it's completely the opposite. So a plant with aphids, they were attracted to that because that's their prey. They're attracted to it. But also the plants which were linked up with the mycorrhizal connections, they were also attractive. But there wasn't any attraction to plants where there wasn't that connection. So that was a really fascinating study. It's more fundamental, but it shows the kind of complexity of the signaling that occurs in nature through the mycorrhizae, and the key chemical that was involved in that change in the smell of the plants was methyl salicylate, because what we did was we had some samples which were attractive, and if you get the amount of the methyl, because that was the only compound that seemed to have a significant difference, but if you get the same amount of it that's present in a repellent sample, and you spike the attractive one with a bit of that, it switches off the attraction. So there's the attractive sample to begin with, and then if you add the methyl salicylate, then it becomes repellent. So that was the key compound within the context of that blend that was carrying that signal. But I've also done a lot of work on this topic, looking at maize plants with subsistence farming in Africa. So are the plants with eggs on them more attractive to these parasitic wasps? And this is the kind of damage that these stem borer moths can do. There's another pest now that we've got a new project on, the fall armyworm, that's also wreaking havoc there, but which way we're going to look at similar things. But the hypothesis is that when the plant's got eggs on it, it releases a different odour that attracts the natural enemies. And also that the farmer preferred land races, because they've been selected under lower input conditions, that they might actually have better stress resistance than some of these plants which have been grown in very very cushy environments where they've had a high input regime to protect them from pests and application of a lot of fertilizers and things. So are the farmer-preferred varieties more stress-resistant? In particular, can you get improved attraction to the plants with eggs on them? So we get the plants, put them in these cages overnight, so with or without the moths, and the ones which have got the moths, they get the eggs, and then collect the odours from those plants using this portable odour collection. This is just one. There were many of these that were done. And collecting the odours and then doing that olfactometer bioassay. So this has all been set up in Issappi in Kenya. And then the vast majority of the commercial hybrids don't have that trait of attracting the natural enemies. But the interesting thing was that most of the land races, particularly the farmer-preferred ones, that they were attractive to natural enemies. So it's a kind of indirect defence, this cry for help. And it's at the very earliest stage of herbivory, so right when the insect is laying her eggs on the plant. And the interesting thing was that out of those commercial hybrids, the ones that did have that trait actually came from a different type of breeding program. So this came from Seedco, and they were specifically growing varieties for the African smallholder farmer market where they didn't have such high input levels. So that was seemed that it was potentially an explanation for this. And there's also some early field data showing that there's increased parasitism under real field conditions, so it's not just in the lab. We've also been exploring the possibility of using the aggregation pheromone of this pea and bean weevil to improve the spread of this entomopathogenic fungus. So developing these killing stations which have got the pheromone to attract the insect, and this is a very useful pheromone because it attracts the females as well as the males, and trying to spread around this entomopathogenic fungus. But this is still very experimental and still under evaluation. But this is the kind of killing station. But and it works very well, the entomopathogenic fungus for killing the insects. And this is the pheromone, which is very attractive. But it's how to put it all together, and so it's quite ambitious because you want to go away from spraying the whole field, blanket spraying with insecticide, and then instead getting the insects to come to you, to come into this little device, and then go back out again and spread this thing around. But At the moment, there don't seem to be enough of them that are going through, but it's still quite experimental, that. But so the final section is agroecology, and we're going to look at this push-pull example. So the stem borers, this is again with the subsistence farmers in sub-Saharan Africa, and stem borers cause huge yield losses, and also this striga weed, which is a parasitic plant. This is a terrible situation. I mean, trying to pull it up by hand, it's already done all the damage there, but it causes massive yield losses and really cripples the crops. But the good news, though, is that there's this really neat companion cropping system which was developed actually with Lester Wadhams, who's in the audience today, but in collaboration with ISIPI in Africa. So it's Rothamsted and ISIPI collaborating, and ISIPI is based in Kenya, and it's got these trap crops around the edge of the plant. So that's the pull part of the push-pull, and then the push part of it is these intercrops which are releasing repellents. And so although it's a very low-tech kind of approach which could be used by smallholder farmers, there's a lot of science which is underpinning it, because you can't just bang in any crop there. You need to have the right ones which are releasing the right chemistry. So you need to have a plant which is releasing repellents in between the crops and a trap crop that's attractive around the edge. And a wonderful spin-off from this was that some of the root exudates from one of the intercrops, it could suppress the striga. So it caused suicidal germination of the striga. So that was a wonderful development, because not only is it managing the insect pest, which is how it got this funny name of the push-pull, but it's also suppressing the weeds. And so that was fantastic to get that. And it really makes a tremendous difference, because here you have the monocrop maize with the striga weed, and it really stunts the growth of the plant. And then at the same place, same growing conditions, same maize variety, everything, but you've got this other plot. And the only difference is that it's got this desmodium in between, and you can see how much bigger, how much healthier these plants are, and they've got some proper cobs there that's going to provide more of a harvest for the farmers. So that's fantastic. It's a really wonderful example, and it's a privilege to have been involved in that collaboration. But as I say, it needs to have the right chemistry. You can't just put any plant in. You need to have the right plants which are releasing the right chemicals. And that's what it looks like at two different stages. This is early in the season. You can see the napier grass around the edge, and you can see the desmodium starting there. And this is once the crop has grown bigger later on. And the bottom line is the yield, and it doubles or triples the yield. So it makes a tremendous difference, and that's because the farms can be really badly affected by these pests and things. So just rounding up now, and so final couple of slides, and so we're sort of zooming back out again now. And so here we've got this pristine environment. It's the Peruvian Amazon, and here we've got we've got nature there, and there's leaching of the tannins from the decaying leaves of the adjoining vegetation. So that explains why this black water stream. So you see there's two different sorts of streams here. There's a black water stream and a white water stream. And the black water stream, it's hardly got anything living in it, because it's so acidic. It's got the water chemistry inhibits the growth of insect larvae or pretty much anything, and it's almost sterile because the pH is so low, and so this is actually plant defense in nature, and that's the kind of effect that's happening there. And so I think we need to think about these things and what happens in nature and what happens in farming, and we don't necessarily want to take everything that's in nature, because some natural compounds can actually be highly toxic. Take, for example, the mycotoxins. Those can be really harmful to human health, and so I don't think it's a rational point of view to say that any chemical which is natural is safe, because obviously you wouldn't send your children off into the woods to eat anything they see there. So you need to have the right. You need to think about these things, but try and get something which is bad for the insects but good for the people. And for plants, I mean, the plant defense, they're rooted to the ground, and so they have to evolve some pretty sophisticated ways of surviving when they're literally fixed to the ground, because I mean, if someone tries to attack me, I can run off, that's it, but I can go. But if I get stage fright here, I can just quickly go away. But a plant hasn't got that option, it's so they have needed to evolve some pretty sophisticated things to allow themselves to survive, and I think we can learn from that. And so and if you go back to the roots of the environmentalist movement, and Rachel Carson in Silent Spring, she was actually very pro-science. So this is what she said. We need to have more biological solutions, and we need to have entomologists, pathologists, geneticists, physiologists, biochemists, ecologists, all working together to provide a new science of biotic controls. And I think she was actually ahead of her time, and that's the kind of thing that we're trying to do at Keele here now with some of the new research in the group that I'm so fortunate to be leading now. But there is a major challenge. One of the last projects that I did at Rothamsted was a bit different, because it was translational rather than original research, and we were trying to share management recommendations with farmers about how to manage pests, weeds and diseases. And the really interesting about this was because we developed a mobile-friendly phone app that they could use for looking up this information. But the nice thing about it was that you could get information in the other direction and find out what their priorities were. And all the way through, we were trying to ask them, what is important to you? What are the targets that matter? And so this isn't the scientists speaking. This is actually coming from the network of farmers. So there was over 1,000 farmers and agronomists who were members of this thing. And this was the kind of crowdsourced reporting of what their problems are. And so right at the top of the list are things where you've got problems with pesticide resistance, or where a pesticide has recently been withdrawn. So blackgrass, there's huge issues with herbicide resistance there. Slugs, we've lost some of the treatments that used to be used there. Cereal aphids and particularly this cabbage stem flea beetle. Now, you're probably aware of the neonicotinoid restriction, because that was banned as a precautionary measure to protect the bees. But it's left a huge hole in the crop protection armoury, because this flea beetle is now devastating the very young oilseed rape crops, and so the farmers are having some really big problems with some of these things. And it's what we need to do is, we need to increase the pace of innovation, because at the moment what's happening is the speed at which new solutions are coming through to the farmer is slower than the speed at which the pests are evolving resistance to these things. So we need to increase the pace of innovation so that we can get some new solutions that are actually useful for the farmers. And this is a lot easier said than done. But because there's so many things that we want to make sure that the interventions are safe and everything as well. But there is an urgent need to increase this, because if you consider that the farmer's got so many problems right now, and we're not just talking about niche organic farming now, we're talking about major crops like wheat, you can't grow it because of blackgrass, or oilseed rape, you can't grow it because of the flea beetle. So we need some new solutions pretty rapidly, and you also need to consider that that it takes take 10 years to get something from concept to field delivery. So those problems are only going to get worse as time goes on. So we need to increase the pace of innovation, basically. But that's so thank you very much for listening. It's been, I'm really honoured to have everyone come here in the audience this evening. I really appreciate it. And I'd also like to thank everyone, because obviously some of this research that I'm been describing, it's not just me on my own, it's been a collaborative effort, and there's so many people who've been involved in it. I can't name everyone, but there's some of my former colleagues here at Rothamsted and PhD students, Ben Webster, Zdenka Babikova, Christine Wilcox, she's a really good physiologist, Leslie Smart and Janet Martin, they're really good field entomologists, and then John Pickett and Lester Wadham, so my former senior leaders at Rothamsted. And here at Keele, we're doing some really nice research with Claire Hoskins and developing these nanoformulations, and Joe Roberts, David Buss and William Kirk. So our group is growing, and there's an exciting time ahead. And of course the collaboration with ISIPI in Kenya has been really a pleasure to be involved with things like the push-pull with Zia Khan, Charles Bodega and Emmanuel Tamira. So thank you so much for listening. I think there may be some time for questions if anyone has any, but thank you.